Hello, I am Dr. Owen On, and welcome to the next installment of the Boring Lecture series. In this lecture, we will be exploring the thermoregulatory and biochemical responses of sheep and goats to natural saline water. Today's paper of interest is a localized study, evaluating these responses under real-world pastoral conditions in the Mid-Rift Valley of Ethiopia. The interaction between environmental stressors and physiological responses in livestock has long been a subject of investigation within the fields of animal physiology, veterinary science, and pastoral systems research. Among these environmental stressors, water quality occupies a central position due to its fundamental role in maintaining homeostasis, metabolic function, and thermoregulation. In arid and semi-arid regions of the world, where pastoralism is the dominant livestock production system, access to high-quality fresh water is often limited. Consequently, animals frequently consume water containing elevated concentrations of dissolved salts. The study under consideration, conducted by Chulu et al. 2025, examines the thermoregulatory and biochemical responses of indigenous sheep and goats exposed to naturally saline drinking water under real-world pastoral conditions in the Mid-Rift Valley of Ethiopia. The importance of this research lies not only in its physiological findings, but also in its relevance to climate resilience, livestock adaptation, and sustainable pastoral livelihoods. To fully appreciate the scope and implications of this study, it is necessary to first define, in explicit and exhaustive detail, the core concepts involved. These include thermoregulation, biochemical parameters, water salinity, pastoral production systems, and species-specific adaptive traits in sheep and goats. The following sections will therefore provide a deliberately extensive and detailed background, including definitions of commonly used terms that may otherwise be assumed to be self-evident. Thermoregulation refers to the physiological processes by which an organism maintains its core body temperature within a narrow, optimal range, despite fluctuations in environmental temperature. In mammals, including sheep, ovisaries, and goats, Capra hircus, thermoregulation is achieved through a combination of behavioral, anatomical, and physiological mechanisms. Physiological thermoregulation involves processes such as alteration of respiratory rate, peripheral vasodilation and vasoconstriction, sweating or panting, and modulation of metabolic heat production. In livestock species, thermoregulation is particularly important because deviations from the thermal neutral zone can lead to heat stress or cold stress, both of which have measurable impacts on productivity, immune function, and metabolic efficiency. Within the context of the present study, thermoregulation is primarily assessed through rectal temperature, respiratory rate, and pulse rate. Each of these parameters serves as an indirect indicator of the animal's ability to dissipate or conserve heat under environmental and dietary stress conditions, including exposure to saline drinking water. It is worth emphasizing that thermoregulation is not a single mechanism, but rather a complex integration of multiple physiological systems working in concert. The hypothalamus serves as the primary regulatory center, receiving input from peripheral and central thermoreceptors and coordinating appropriate effector responses. These responses may be autonomic, such as changes in cutaneous blood flow, or behavioral, such as seeking shade or altering posture. The thermal neutral zone, previously mentioned, represents the range of ambient temperatures within which an animal can maintain its body temperature through minimal adjustments in metabolic rate. When ambient temperatures exceed the upper critical temperature of this zone, animals must employ active cooling mechanisms. Conversely, temperatures below the lower critical temperature require increased heat production through metabolic activity. Rectal temperature is defined as the temperature measured within the rectum, typically using a digital or mercury thermometer inserted to a standardized depth. It is widely regarded as a reliable proxy for core body temperature in domestic animals. In small ruminants, normal rectal temperature ranges are generally reported as sheep, approximately 38.3 to 39.9 degrees Celsius, and goats, approximately 38.5 to 40.0 degrees Celsius. Variations within these ranges may occur due to factors such as ambient temperature, physical activity, time of day, and water intake and composition. Elevated rectal temperatures may indicate thermal load, dehydration, or impaired heat dissipation, while reduced temperatures may reflect cold exposure or metabolic suppression. In the present study, rectal temperature is used as a primary indicator of thermoregulatory strain associated with saline water consumption. The measurement of rectal temperature, while seemingly straightforward, requires careful attention to procedural standardization. The thermometer must be inserted to a consistent depth to ensure reproducibility of measurements. Typically, insertion depths of 8 to 10 centimeters are employed in adult small ruminants, though this may vary depending on animal size. The position of the thermometer bulb relative to the rectal mucosa is also critical. Contact with fecal material or air pockets within the rectum may yield artificially low or variable readings. Therefore, the thermometer should be positioned such that the sensing element is in direct contact with the rectal wall. Time of day represents another important consideration in the interpretation of rectal temperature measurements. Circadian rhythms influence core body temperature in most mammals, with temperatures typically being lower in the early morning hours and higher in the late afternoon. This diurnal variation must be accounted for when comparing measurements taken at different times of day. Furthermore, recent physical activity can transiently elevate rectal temperature due to increased metabolic heat production. In pastoral systems where animals may walk considerable distances to access water and forage, this factor assumes particular relevance. Respiratory rate refers to the number of breaths taken per unit time, typically expressed as breaths per minute. In ruminants, respiration plays a dual role in gas exchange, encompassing oxygen uptake and carbon dioxide elimination, and heat dissipation through evaporative cooling. Under conditions of heat stress, respiratory rate often increases as animals engage in panting, a mechanism that enhances evaporative heat loss from the respiratory tract. However, prolonged elevations in respiratory rate may lead to respiratory alkalosis, particularly if accompanied by excessive loss of carbon dioxide. In sheep and goats, normal resting respiratory rates typically range from 20 to 40 breaths per minute, depending on species, age, and environmental conditions. In this study, changes in respiratory rate are interpreted as indicators of both thermal stress and physiological compensation in response to saline water ingestion. The mechanism of panting deserves additional elaboration. Panting involves rapid, shallow breathing that increases airflow over the moist surfaces of the upper respiratory tract, particularly the tongue, oral cavity, and nasal passages. Evaporation of water from these surfaces removes heat from the body, thereby contributing to thermoregulation. However, panting is not without physiological cost. The increased work of breathing requires energy expenditure, and the associated hyperventilation can disturb acid-base balance. When carbon dioxide is eliminated at a rate exceeding its metabolic production, blood pH increases, resulting in respiratory alkalosis. This condition may be partially compensated through renal mechanisms, but prolonged panting can nonetheless impose a significant physiological burden. The efficiency of evaporative cooling through panting is influenced by ambient humidity. In environments with high relative humidity, the capacity for evaporation is reduced, thereby limiting the effectiveness of this thermoregulatory mechanism. Conversely, in arid environments characteristic of many pastoral systems, low humidity facilitates evaporative cooling but may simultaneously increase the risk of dehydration. Species differences in reliance on panting versus other cooling mechanisms exist. Goats, for instance, are often reported to have more efficient cutaneous evaporative cooling compared to sheep, which may reduce their dependence on respiratory heat dissipation under certain conditions. Pulse rate, also referred to as heart rate, is defined as the number of cardiac contractions per minute. It reflects the combined effects of metabolic demand, cardiovascular efficiency, and thermoregulatory adjustments. Increases in pulse rate may occur due to elevated metabolic heat production, peripheral vasodilation during heat stress, and dehydration-induced reductions in plasma volume. Typical resting pulse rates in small ruminants are approximately sheep 70 to 90 beats per minute and goats 70 to 110 beats per minute. Monitoring pulse rate allows researchers to assess the cardiovascular cost of maintaining homeostasis under environmental and nutritional stresses. The relationship between pulse rate and thermoregulation warrants further examination. During heat stress, peripheral vasodilation occurs to facilitate heat transfer from the body core to the skin surface, where it can be dissipated to the environment through radiation and convection. This vasodilation results in a redistribution of blood flow, with a greater proportion of cardiac output directed to cutaneous vessels. To maintain adequate tissue perfusion in the face of this redistribution, heart rate typically increases. The magnitude of this increase reflects both the severity of the thermal challenge and the efficiency of other thermoregulatory mechanisms. Dehydration, which may occur as a consequence of inadequate water intake or excessive water loss, further complicates cardiovascular function during heat stress. Reduction in plasma volume decreases venous return to the heart, potentially compromising stroke volume. To maintain cardiac output in the presence of reduced stroke volume, heart rate must increase proportionally. The measurement of pulse rate in small ruminants is typically accomplished through palpation of accessible arteries, most commonly the femoral artery located on the medial aspect of the hind limb. Alternatively, auscultation of the heart using a stethoscope placed over the left thorax may be employed. As with rectal temperature and respiratory rate, pulse rate exhibits diurnal variation and is influenced by factors such as recent physical activity, excitement, and ambient temperature. Standardization of measurement conditions is therefore essential for meaningful interpretation of results. Let us now examine the biochemical parameters measured. Biochemical parameters refer to measurable chemical constituents of blood or serum that provide insight into metabolic status, organ function, hydration, and electrolyte balance. In the study by Chulu et al. 2025, the biochemical parameters assessed include, but are not limited to, serum sodium, serum potassium, serum chloride, total protein, glucose, urea, and creatinine. Each of these parameters serves a distinct physiological role and responds predictably to changes in water intake, salinity, and hydration status. The measurement of biochemical parameters in blood provides a window into the internal physiological state of an animal that cannot be assessed through external observation alone. While thermoregulatory parameters such as rectal temperature and respiratory rate reflect the animal's immediate response to environmental conditions, biochemical measurements reveal the underlying metabolic and homeostatic adjustments occurring at the cellular and systemic levels. Blood, as the primary transport medium for nutrients, metabolic wastes, hormones, and electrolytes, serves as an integrative indicator of physiological status. Changes in the concentration of blood constituents may reflect alterations in their production, utilization, distribution, or elimination. The interpretation of biochemical parameters requires consideration of multiple factors. Normal reference ranges vary among species, breeds, ages, and physiological states. Additionally, sample collection and handling procedures can influence measured values. Hemolysis, for example, can artificially elevate serum potassium concentrations due to release of intracellular potassium from damaged red blood cells. The timing of blood collection relative to feeding, water intake, and environmental stressors may also affect results. For this reason, standardized protocols specifying the time of day and animal activity level at the time of sampling are essential for generating reproducible and interpretable data. Water salinity refers to the concentration of dissolved inorganic salts present in water. Salinity is commonly measured using several interrelated parameters. Electrical conductivity is a measure of water's ability to conduct an electric current, which increases with higher concentrations of dissolved ions. EC is typically expressed in microsiemens per centimeter or millisiemens per centimeter. Higher EC values indicate higher total dissolved salts, though EC does not identify specific ions present. The principle underlying electrical conductivity measurement is based on the fact that ionic substances dissociate in aqueous solution to produce charged particles that facilitate the flow of electrical current. The magnitude of current flow under a standardized electrical potential is proportional to the concentration of dissolved ions. It should be noted that electrical conductivity provides an aggregate measure of ionic content but does not distinguish among different types of ions. Water containing high concentrations of sodium chloride will exhibit similar conductivity to water containing equivalent ionic concentrations of other salts, despite potentially different biological effects. Temperature significantly affects conductivity measurements, as ionic mobility increases with temperature. For this reason, conductivity meters typically include automatic temperature compensation or require manual correction of readings to a standard reference temperature, usually 25 degrees Celsius. Total dissolved solids represent the combined concentration of all inorganic and organic substances dissolved in water. TDS is commonly expressed in milligrams per liter. TDS values are often estimated from EC using a conversion factor, though this factor varies depending on water composition. The relationship between EC and TDS is not universal but depends on the specific ionic composition of the water being analyzed. In many freshwater systems, a conversion factor of approximately 0.6 to 0.7 is used, meaning that TDS in milligrams per liter equals EC in microsiemens per centimeter multiplied by this factor. Direct measurement of TDS can be accomplished by evaporating a known volume of water to dryness and weighing the residual solids. However, this method is more time-consuming than conductivity measurement and is therefore less commonly employed in routine water quality assessment. The biological significance of TDS lies in its effect on water palatability and osmotic stress. High TDS concentrations can reduce voluntary water intake in livestock, potentially leading to dehydration. Additionally, the osmotic gradient created by dissolved solutes affects cellular hydration and electrolyte balance. pH is a logarithmic measure of hydrogen ion concentration, indicating the acidity or alkalinity of water. While pH does not directly measure salinity, it influences ion solubility and palatability of drinking water. The pH scale ranges from 0 to 14, with 7 representing neutrality. Values below 7 indicate acidic conditions, while values above 7 indicate alkaline conditions. Because the scale is logarithmic, each unit change in pH represents a tenfold change in hydrogen ion concentration. In the context of livestock water quality, pH affects several important processes. Extreme pH values can directly irritate mucous membranes of the gastrointestinal tract. Additionally, pH influences the solubility and bioavailability of minerals present in water. Some toxic metals become more soluble and therefore more bioavailable at low pH, while certain essential minerals may precipitate out of solution at high pH. The pH of natural water bodies is influenced by geological factors, including the mineral composition of surrounding soils and rocks, as well as biological processes such as photosynthesis and respiration by aquatic organisms. In closed basin lakes with no outlet, evaporative concentration of dissolved salts often leads to elevated pH values. We shall now look at the pastoral production systems. A pastoral system is a livestock production system characterized by extensive grazing, seasonal mobility, and reliance on natural forage and water sources. In Ethiopia's Mid-Rift Valley, pastoral and agripastoral communities depend heavily on surface water sources, many of which are naturally saline due to geological and hydrological factors. Animals in these systems are therefore chronically exposed to environmental conditions that differ substantially from controlled experimental settings, making field-based studies such as this one particularly valuable. Pastoral production systems represent one of the oldest forms of livestock husbandry and remain critically important for the livelihoods of millions of people worldwide, particularly in arid and semi-arid regions where crop production is not viable. These systems are characterized by their adaptive nature, with livestock management strategies evolved over generations to cope with variable and often harsh environmental conditions. The extensive nature of pastoral grazing means that animals range over large areas in search of forage and water. Stocking densities are typically low compared to intensive production systems, and animals subsist primarily on natural vegetation rather than cultivated crops or supplemental feeds. Seasonal mobility, also known as transhumance in some contexts, involves the movement of livestock herds between different grazing areas in response to seasonal variations in forage availability and water access. During wet seasons, pastoralists may utilize grazing lands distant from permanent water sources, while dry seasons often necessitate concentration around reliable water points. The reliance on natural resources makes pastoral systems vulnerable to environmental variability and change. Droughts, which occur with variable frequency in arid regions, can drastically reduce forage availability and water access, leading to livestock losses and food insecurity. Climate change is expected to exacerbate these challenges through increased frequency and severity of extreme weather events. Despite these vulnerabilities, pastoral systems demonstrate remarkable resilience when traditional management practices are maintained and when access to key resources is not restricted. The mobility that characterizes these systems allows for tracking of spatially and temporally variable resources, thereby buffering against local environmental variability. We shall now examine species differences between sheep and goats. Sheep and goats, while both classified as small ruminants, differ in several physiological and behavioral traits relevant to water salinity tolerance. Goats are generally regarded as more adaptable to harsh environments, more selective feeders, and more tolerant of dietary and water-related stressors. Sheep, by contrast, are often considered less tolerant of high salinity, more susceptible to dehydration, and more sensitive to electrolyte imbalances. The present study repeatedly compares these two species to assess differential adaptive responses under identical environmental conditions. These species-level differences have been documented across numerous studies and geographic regions. Goats, in particular, have long been recognized for their ability to thrive in marginal environments where other livestock species struggle. This resilience is attributed to several factors, including behavioral, anatomical, and physiological adaptations. Behaviorally, goats exhibit greater selectivity in their feeding compared to sheep. While sheep are predominantly grazers, consuming primarily grasses and low-growing herbaceous vegetation, goats are browsers that preferentially consume leaves, shoots, and bark from woody plants. This dietary flexibility allows goats to exploit resources that may be unavailable or unpalatable to other species. The browsing behavior of goats also affects their water requirements. Woody vegetation tends to have higher moisture content than dried grasses, potentially reducing the need for drinking water during periods when forage is available. Additionally, the metabolic water produced during digestion of dietary dry matter contributes to total water balance. Anatomically, goats possess features that may enhance their ability to cope with water scarcity and heat stress. Their coat characteristics differ from those of most sheep breeds, typically being shorter and less insulative. This may facilitate heat dissipation, reducing the thermal load that must be managed through evaporative cooling. Physiologically, goats demonstrate superior ability to concentrate urine, thereby conserving water during periods of limited availability. The kidneys of goats can produce urine with higher osmolality than that of sheep, allowing for maintenance of hydration status with less water intake. Goats also exhibit different patterns of water intake compared to sheep. Under conditions of water restriction, goats may consume larger volumes when water is made available, compensating for periods of deprivation. This ability to rapidly rehydrate may contribute to their tolerance of intermittent water access. The differences in salinity tolerance between sheep and goats likely reflect the combined effects of these and other adaptations. However, it should be noted that considerable variation exists within species, with some breeds of sheep exhibiting greater salt tolerance than others. Indigenous breeds adapted to arid environments may possess traits that enhance their resilience to saline water consumption. We shall now examine the site. The study conducted by Chulu et al. 2025 was carried out in the Mid-Rift Valley region of Ethiopia, a geographically and ecologically distinct area that forms part of the larger East African Rift system. This region is characterized by a combination of semi-arid climatic conditions, volcanically influenced geology, and a long-standing tradition of pastoral and agripastoral livestock production. The inclusion of this study area is not incidental. Rather, it reflects the real-world conditions under which indigenous sheep and goats are maintained and managed, thereby increasing the ecological validity of the findings. The Mid-Rift Valley represents a region of considerable importance both ecologically and economically. The area supports diverse human populations engaged in various livelihood strategies, with pastoralism and agripastoralism predominating in many areas. The presence of permanent and seasonal water bodies, including lakes, rivers, and streams, makes the region relatively more favorable for livestock production compared to more arid zones. However, these water resources vary considerably in quality, with many exhibiting elevated salinity due to the geological characteristics of the region. Understanding how livestock cope with the consumption of these naturally saline waters is therefore of direct practical relevance to the communities who depend on these animals for their livelihoods. The study by Chulu et al. Was conducted under naturally occurring conditions, meaning that animals were exposed to seasonal dynamics without experimental manipulation. The study references naturally saline water sources commonly utilized by pastoral livestock. These include shallow wells, ponds, streams, and lakes and lake-adjacent watering points. One notable example frequently cited in the literature, though not necessarily the sole focus of the study, is Lake Basaka, a saline lake whose expansion and increasing salinity have been documented over several decades. Such water sources are often the only available options for livestock during periods of water scarcity. Livestock tolerance to saline water varies by species, physiological state, and duration of exposure. Goats are often reported to tolerate higher salinity levels than sheep, a distinction that underpins the comparative nature of this study. The sheep and goats included in the study are described as indigenous, meaning they are locally adapted breeds that have evolved or been selectively maintained under the environmental conditions of the region. Local adaptation may include enhanced tolerance to heat, improved ability to concentrate urine, behavioral strategies to minimize water loss, and altered electrolyte handling. These adaptive traits are of particular interest when assessing responses to saline water under pastoral conditions. Indigenous breeds represent populations that have been subjected to natural and artificial selection under local environmental conditions over many generations. The result is a suite of adaptations that enhance fitness in the specific ecological context where the breed has evolved. In the case of small ruminants in arid and semi-arid regions of Ethiopia, selection pressures have likely favored traits that enhance survival and reproduction under conditions of limited water and forage availability, high ambient temperatures, and exposure to endemic diseases and parasites. Some of these adaptive traits are visible externally, such as coat color and hair versus wool covering. Others are physiological and require experimental investigation to characterize fully. The study by Chulu et al. Contributes to understanding of these physiological adaptations by documenting the responses of indigenous animals to naturally saline water. It is important to recognize that local adaptation comes at potential costs. Breeds specialized for particular environments may perform poorly when moved to different environments. Additionally, intensive selection for specific traits may reduce genetic diversity and limit the population's capacity to respond to novel selection pressures. Conservation of indigenous breeds is therefore important not only for maintaining current pastoral production systems, but also for preserving genetic resources that may be valuable under future environmental conditions. The environmental context described above directly influences thermoregulatory parameters measured in the study. Elevated ambient temperatures increase the reliance on respiratory evaporative cooling, peripheral vasodilation, and behavioral modifications, such as reduced activity during peak heat. When combined with saline water intake, these thermoregulatory demands may place additional strain on physiological systems. Thermoregulation does not occur in isolation from other physiological processes but is intimately linked to energy metabolism, water balance, and electrolyte homeostasis. The consumption of saline water affects multiple aspects of this integrated system. High salt intake increases osmotic load, requiring the kidneys to excrete concentrated urine to eliminate excess electrolytes. This concentrating process consumes energy and may increase heat production, thereby adding to thermal burden. Additionally, the production of concentrated urine requires adequate water availability. If water intake is insufficient to support both urinary excretion and evaporative cooling, the animal faces a physiological dilemma. The behavioral thermoregulatory strategies available to animals in pastoral systems are constrained by management practices and resource distribution. While animals may seek shade during the hottest part of the day, the availability of shaded areas varies across the landscape. Trees and shrubs that provide shade are often concentrated in certain locations, and movement to these areas may require additional energy expenditure. Reduction of physical activity during hot periods represents another behavioral strategy. However, in pastoral systems where animals must walk to access water and forage, the option to remain inactive is limited. Herders may adjust the timing of animal movements to avoid peak heat, but the spatial distribution of resources may necessitate travel during hot hours. We shall now transition to the experimental design. The study conducted by Chulu et al. 2025 employed an observational field-based experimental design aimed at evaluating the physiological and biochemical responses of indigenous sheep and goats consuming naturally saline water under pastoral management conditions. Unlike controlled laboratory experiments, this design was selected to reflect the realistic environmental and management conditions experienced by animals in the Mid-Rift Valley of Ethiopia. The animals included in the study consisted of indigenous sheep and goats maintained by pastoral households within the study area. Selection criteria were applied to ensure relative uniformity while preserving ecological relevance. 2 species were included: sheep, taxonomically Ovis aries, and goats, taxonomically Capra hircus. The inclusion of both species enabled a comparative assessment of responses to saline water consumption, reflecting known differences in adaptive capacity between sheep and goats. The decision to include both species in the study design was based on their co-occurrence in pastoral production systems of the region and their documented differences in environmental tolerance. By measuring responses in both species under identical environmental conditions, the study provides insight into species-specific adaptation mechanisms. Animals selected for the study were adults, as determined by dentition and owner records. The use of adult animals reduced variability associated with growth-related metabolic changes, immature thermoregulatory systems, and developmental differences in renal function. The criterion of adult age is important because young animals differ from adults in numerous physiological characteristics that could confound interpretation of results. Growing animals have higher metabolic rates per unit body mass than adults, affecting thermoregulation. Additionally, the kidneys of young animals may not have reached full functional capacity for urine concentration. Only clinically healthy animals were included. Health status was assessed through visual inspection, observation of gait and posture, evaluation of body condition score, and absence of overt clinical signs such as nasal discharge, diarrhea, or lameness. Animals exhibiting signs of illness were excluded to prevent confounding effects on physiological and biochemical parameters. Clinical assessment of health in field settings relies primarily on physical examination rather than diagnostic testing. Visual inspection can reveal signs of respiratory disease, skin conditions, or systemic illness. Gait observation identifies lameness or neurological abnormalities. Body condition scoring provides an integrated assessment of nutritional status. Body condition score, BCS, was used as an indicator of nutritional status. BCS reflects the amount of fat and muscle reserves present in the animal and is commonly assessed using a standardized scale. In small ruminants, BCS is typically evaluated on a 1 to 5 scale, where 1 indicates emaciation and 5 indicates obesity. Animals selected for the study were generally within the moderate range, thereby minimizing the influence of extreme nutritional states on study outcomes. Body condition scoring involves palpation of specific anatomical sites to assess subcutaneous fat deposition and muscle mass. In small ruminants, the lumbar region is commonly used for this assessment. The prominence of vertebral processes and the degree of fat cover over and around them provide the basis for score assignment. Nutritional status affects numerous physiological parameters. Animals in poor body condition have reduced energy reserves and may be less able to mount effective thermoregulatory responses. Conversely, animals in excessively fat condition have reduced surface area to volume ratio, potentially impairing heat dissipation. By selecting animals within a moderate BCS range, the researchers minimized this source of variability, making it more likely that observed differences among groups reflected water source effects rather than nutritional differences. Animals were grouped based on species, whether sheep or goat, access to specific water sources, and exposure to saline drinking water. The grouping strategy ensured that observed differences could be attributed primarily to species-specific physiological responses rather than differences in water availability or management practices. In the study design, animals were categorized into groups based on the water source they utilized. One group consisted of animals that consumed water from Lake Basaka, representing the saline water treatment. Another group consumed water from the Awash River or other freshwater sources, serving as a control or comparison group. Within each water source category, animals were further divided by species, creating a factorial design with water type and species as the two main factors. This design allows for assessment of main effects of each factor as well as potential interaction effects. An interaction between water type and species would indicate that the response to saline water differs between sheep and goats. Such an interaction would be consistent with the hypothesis that goats possess greater tolerance to saline water than sheep. The water sources utilized by the animals were naturally saline, consistent with the hydrological characteristics of the Mid-Rift Valley. Water samples were collected from watering points used by the experimental animals. Sampling procedures included collection in clean, labeled containers, avoidance of sediment disturbance, and transportation to the laboratory under appropriate conditions. Samples were analyzed for electrical conductivity, total dissolved solids, pH, and major ions such as sodium and chloride. Electrical conductivity was measured using a calibrated conductivity meter. Calibration procedures were performed prior to measurement using standard solutions to ensure accuracy. EC values provided an indirect estimate of total ionic concentration and were used to classify water salinity levels. Conductivity meters function by applying a voltage across two electrodes immersed in the water sample and measuring the resulting current flow. The magnitude of current is proportional to the concentration of ions in solution. Total dissolved solids were either measured directly or estimated from EC values using established conversion factors. The resulting TDS values were expressed in milligrams per liter. These values served as a quantitative indicator of the salinity burden imposed on the animals through drinking water. TDS can be measured gravimetrically by evaporating a known volume of water and weighing the residue. However, this method is time-consuming and requires access to analytical balances and drying ovens. Estimation of TDS from EC measurements provides a more rapid alternative, though the accuracy of this estimation depends on the validity of the conversion factor used. The conversion factor varies depending on the ionic composition of the water, so site-specific calibration may improve accuracy. For pastoral water quality assessment, the precision afforded by gravimetric TDS measurement may not be necessary, and EC-based estimates may be adequate for categorizing water sources as fresh, slightly saline, moderately saline, or highly saline. Water pH was measured using a digital pH meter. pH measurements were conducted at ambient temperature, and the meter was calibrated using standard buffer solutions. Although pH does not directly quantify salinity, it influences water palatability, ion solubility, and potential interactions with gastrointestinal physiology. pH meters measure the electrical potential difference between a pH-sensitive electrode and a reference electrode. The magnitude of this potential difference is related to hydrogen ion activity in the solution according to the Nernst equation. Thermoregulatory parameters were measured using standardized procedures to ensure consistency. Rectal temperature was measured using a digital veterinary thermometer. The thermometer was inserted to a consistent depth, held in place until a stable reading was obtained, and cleaned between animals to prevent cross-contamination. Measurements were typically taken during the daytime, under ambient conditions reflective of normal pastoral activity. The procedural details of rectal temperature measurement warrant additional attention. Insertion depth affects the temperature recorded, as temperature may vary along the length of the rectum. Deeper insertion generally provides more stable readings that better reflect core body temperature. Animal restraint during temperature measurement should be sufficient to allow safe insertion and positioning of the thermometer, but should minimize stress that might artificially elevate body temperature. Experienced handlers can typically accomplish this efficiently. Respiratory rate was measured by visually observing flank movements. The number of breaths was counted over a fixed time interval, usually 1 minute. Observers were trained to minimize disturbance, as excitement or handling stress can artificially elevate respiratory rate. Visual observation of respiratory rate requires practice to reliably identify individual breaths, particularly at high respiratory rates when flank movements may be rapid and shallow. Each complete respiratory cycle consists of one inspiration and one expiration. Animal disturbance must be minimized because the act of approaching and observing animals can alter their behavior. Observations should be conducted from a distance when possible, using binoculars if necessary, or after allowing animals time to acclimate to the observer's presence. Pulse rate was measured by palpation of accessible arteries, such as the femoral artery. Beats were counted over a standardized time period and converted to beats per minute. Pulse rate measurements provided insight into cardiovascular adjustments associated with thermoregulation and hydration status. The femoral artery is located on the medial aspect of the hind limb and can be palpated in most small ruminants, though ease of palpation varies with body condition and fleece characteristics. With experience, handlers can reliably locate and count the pulse. Alternative methods for measuring heart rate include cardiac auscultation using a stethoscope and use of electronic heart rate monitors. However, these approaches may be less practical under field conditions. Blood samples were collected via jugular venipuncture using sterile needles and collection tubes. Procedural steps included proper animal restraint, site disinfection, collection of an appropriate volume of blood, and immediate labeling of samples. Samples were transported to the laboratory for biochemical analysis. Jugular venipuncture is the standard method for blood collection in small ruminants due to the accessibility and size of the jugular vein. The animal must be properly restrained to prevent movement during needle insertion, which could result in injury or failed collection. Serum samples were analyzed for a range of biochemical parameters using standard laboratory techniques. These analyses provided quantitative data on electrolyte balance, renal function, and metabolic status. We shall now shift to the biochemical evaluation. Biochemical evaluation provides a quantitative framework for assessing internal physiological status in animals exposed to environmental and nutritional stresses. In the context of saline water consumption, biochemical parameters serve as indirect indicators of hydration status, electrolyte balance, renal function, and metabolic regulation. The study by Chulu et al. 2025 included a suite of commonly measured serum biochemical variables. Each of these variables will be described in detail in the following sections, including their physiological roles, normal reference ranges, and expected responses to saline water intake. The measurement of blood biochemical parameters provides information that cannot be obtained through physical examination or observation alone. While external signs may suggest that an animal is coping well with environmental stress, biochemical measurements can reveal subclinical physiological perturbations that may not yet manifest as overt clinical disease. This predictive capability makes biochemical evaluation valuable for assessing animal welfare and for identifying management practices that may pose long-term risks even if short-term effects are minimal. Sodium is the primary extracellular cation in mammals and plays a central role in maintaining osmotic pressure, fluid balance, nerve impulse transmission, and muscle contraction. Serum sodium concentration is tightly regulated by renal mechanisms and hormonal systems, including aldosterone and antidiuretic hormone. Sodium's role as the dominant extracellular cation means that changes in serum sodium concentration affect the osmolality of extracellular fluid. Because water moves freely across most cell membranes to equilibrate osmotic gradients, changes in extracellular osmolality induce water movement into or out of cells. Hypernatremia, defined as elevated serum sodium concentration, creates a hypertonic extracellular environment that draws water out of cells, leading to cellular dehydration. This is particularly problematic for brain cells, as brain tissue is enclosed within the rigid skull and cannot accommodate volume changes without consequences. Conversely, hyponatremia, or reduced serum sodium concentration, creates a hypotonic extracellular environment that drives water into cells, causing cellular swelling. Again, brain swelling is particularly dangerous due to the confinement of the brain within the skull. In small ruminants, normal serum sodium concentrations generally range from 135 to 150 millimoles per liter. Values outside this range may indicate disturbances in water balance or electrolyte regulation. Reference ranges for biochemical parameters should be interpreted with caution, as they are typically derived from populations of healthy animals under specific conditions. Values may vary with breed, age, diet, and environmental factors. Consumption of saline water introduces additional sodium ions into the body. If intake exceeds renal excretory capacity, serum sodium concentrations may increase, leading to hyponatremia. Hyponatremia can result in cellular dehydration, increased thirst, and altered neuromuscular function. The kidneys regulate sodium balance through filtration, reabsorption, and excretion. Under normal conditions, virtually all filtered sodium is reabsorbed, with only a small fraction appearing in urine. When sodium intake increases, the kidneys can increase sodium excretion by reducing the fraction of filtered sodium that is reabsorbed. However, the capacity for sodium excretion is not unlimited. Maximum urine sodium concentration in ruminants is typically lower than serum sodium concentration, meaning that excretion of excess sodium requires production of proportionally more urine. This increases water requirements. If water intake is insufficient to support the production of dilute urine needed to excrete excess sodium, a positive sodium balance develops, and serum sodium concentration rises. This creates a vicious cycle in which high sodium load increases water requirements at the same time that poor water palatability may reduce water intake. Goats are often reported to exhibit greater tolerance to elevated sodium intake due to more efficient renal sodium excretion and enhanced urine concentrating ability. Sheep, by contrast, may exhibit higher serum sodium levels under comparable conditions, reflecting reduced adaptive capacity. The superior salt tolerance of goats has been attributed to several physiological differences. Goats can produce more concentrated urine than sheep, allowing excretion of a given sodium load in a smaller urine volume, thereby conserving water. Additionally, goats may have more efficient systems for sensing and responding to changes in plasma osmolality. The sensitivity of thirst and antidiuretic hormone secretion to osmotic stimuli may differ between species. Behavioral differences may also contribute. Goats may be more willing to consume saline water when no alternative is available, whereas sheep may reduce water intake more severely in response to poor palatability, leading secondarily to dehydration and hyponatremia. Potassium is the primary intracellular cation and is essential for cellular membrane potential maintenance, neuromuscular excitability, and enzymatic function. The distribution of potassium between intracellular and extracellular compartments is maintained by energy-dependent pumps, particularly the sodium-potassium ATPase. Most body potassium is located within cells, where concentrations are typically 30 to 40 times higher than in extracellular fluid. This concentration gradient is essential for establishing the electrical potential across cell membranes. Disruption of potassium homeostasis affects excitable tissues, particularly cardiac muscle and skeletal muscle. Hyperkalemia, or elevated serum potassium, can cause cardiac arrhythmias and potentially cardiac arrest. Hypokalemia, or reduced serum potassium, can cause muscle weakness and altered cardiac function. Typical serum potassium concentrations in small ruminants range from 3.5 to 5.5 millimoles per liter. Serum potassium concentration is maintained within a narrow range under normal conditions despite variations in dietary intake. The kidneys are the primary site of potassium excretion, though some potassium is also lost in feces and sweat. Changes in potassium concentration may occur secondary to altered acid-base balance, renal adjustments, and shifts between intracellular and extracellular compartments. Saline water consumption does not directly increase potassium intake. However, associated dehydration or stress may influence potassium homeostasis. Acid-base disturbances affect potassium distribution. In acidosis, hydrogen ions move into cells and potassium moves out to maintain electroneutrality, potentially causing hyperkalemia. In alkalosis, the opposite occurs, potentially causing hypokalemia. The renal handling of potassium is linked to sodium handling through aldosterone. Aldosterone stimulates sodium reabsorption in the distal nephron and promotes potassium secretion. Therefore, situations that alter aldosterone secretion may affect potassium balance. Cellular injury from any cause can release intracellular potassium into extracellular fluid, causing hyperkalemia. Hemolysis during sample collection or handling is a common artifact that can produce falsely elevated serum potassium values. Goats may maintain more stable potassium levels due to better cellular buffering capacity, while sheep may show greater variability. Data on species differences in potassium homeostasis under saline stress are limited, but general patterns of greater physiological stability in goats compared to sheep may extend to potassium regulation. Chloride is the major extracellular anion and works in concert with sodium to maintain osmotic balance and electroneutrality. Chloride also plays an important role in acid-base balance through the chloride shift, whereby chloride and bicarbonate exchange across red blood cell membranes during oxygen and carbon dioxide transport. Normal serum chloride concentrations typically range from 95 to 110 millimoles per liter. Saline water often contains elevated chloride concentrations, which may lead to increased serum chloride, alterations in acid-base balance, and compensatory renal excretion. Sodium chloride is the predominant salt in many saline waters, meaning that sodium and chloride are ingested in approximately equimolar amounts. The kidneys excrete these ions together, maintaining electroneutrality in urine. Hyperchloremia, or elevated serum chloride, can contribute to metabolic acidosis through mechanisms involving acid-base balance. Normal anion gap acidosis may develop when chloride retention reduces the proportion of bicarbonate in extracellular fluid. As with sodium, goats generally exhibit more effective chloride regulation than sheep, particularly under prolonged exposure. The mechanisms underlying superior chloride handling in goats likely parallel those for sodium, including more efficient renal excretion and potentially different thresholds for hormonal regulation of ion transport. Total protein represents the combined concentration of albumin and globulins. These proteins contribute to oncotic pressure, immune function, and transport of various substances. Serum proteins are synthesized primarily in the liver, with the exception of immunoglobulins, which are produced by plasma cells. Albumin is the most abundant serum protein and is responsible for much of the oncotic pressure that retains fluid within the vascular space. Globulins include a diverse array of proteins with various functions, including transport proteins, clotting factors, and antibodies. The globulin fraction can be further subdivided using protein electrophoresis. Typical serum total protein concentrations in small ruminants range from 6.0 to 8.0 grams per deciliter. Elevated total protein concentrations may reflect hemoconcentration, often associated with dehydration. Conversely, Reduced values may indicate dilution or impaired protein synthesis. Hemoconcentration occurs when plasma volume decreases while the total quantity of protein in the vascular space remains constant, resulting in increased protein concentration per unit volume. This is a common finding in dehydration. Conversely, hemodilution occurs when plasma volume increases relative to protein content, reducing concentration. This may occur with overhydration or with rapid fluid shifts. Changes in total protein concentration due to altered hydration status are typically accompanied by parallel changes in other constituents of blood, including red blood cell concentration as reflected in hematocrit. Sheep may exhibit more pronounced increases in total protein under dehydration, while goats may maintain relatively stable concentrations. The stability of total protein concentration in goats under variable hydration status may reflect their superior ability to maintain plasma volume through more efficient water conservation and utilization. Glucose is a primary energy substrate for many tissues and is tightly regulated through hormonal mechanisms involving insulin and glucagon. Blood glucose concentration is maintained within relatively narrow limits in healthy animals through a balance of glucose absorption from the gastrointestinal tract, glucose production by the liver, and glucose utilization by peripheral tissues. Normal serum glucose concentrations in small ruminants typically range from 50 to 80 milligrams per deciliter. Physiological stress, including heat stress and dehydration, may lead to increased glucose concentrations via cortisol-mediated gluconeogenesis and altered energy metabolism. Cortisol, a stress hormone secreted by the adrenal cortex, stimulates gluconeogenesis, the synthesis of glucose from non-carbohydrate precursors such as amino acids. This increases blood glucose availability to support stress responses. However, chronic stress may deplete glycogen reserves and impair glucose homeostasis, potentially leading to hypoglycemia in severe cases. In ruminants, glucose metabolism differs from that in monogastric animals due to extensive fermentation of dietary carbohydrates in the rumen. Most dietary carbohydrate is converted to volatile fatty acids by rumen microbes, and relatively little glucose is absorbed from the intestine. Ruminants rely heavily on gluconeogenesis to meet glucose requirements, synthesizing glucose primarily from propionate, a volatile fatty acid produced in the rumen, and from amino acids. Goats often display more stable glucose levels under stress, while sheep may show greater fluctuations. The metabolic flexibility of goats may contribute to their ability to maintain glucose homeostasis under variable nutritional and environmental conditions. Urea is a nitrogenous waste product formed in the liver and excreted by the kidneys. It reflects protein metabolism and renal function. Urea is produced through the urea cycle from ammonia, which is generated during amino acid catabolism. In ruminants, ammonia is also produced by rumen microbes from dietary protein and non-protein nitrogen sources. Typical serum urea concentrations in small ruminants range from 20 to 40 milligrams per deciliter. Note that different assays and reporting conventions may express urea as blood urea nitrogen, or BUN, which represents only the nitrogen component of the urea molecule and therefore has lower numerical values. Dehydration associated with saline water intake may reduce renal perfusion, leading to increased serum urea and reduced urine output. When hydration status is compromised, the glomerular filtration rate decreases, reducing the clearance of urea from blood. This results in accumulation of urea in the circulation. Additionally, dehydration stimulates increased urea reabsorption in the renal tubules as part of the urine concentrating mechanism. This further contributes to elevated serum urea. Dietary protein intake also affects serum urea concentration. High-protein diets result in increased amino acid catabolism and greater urea production, while low-protein diets reduce urea production. Sheep may exhibit greater increases in serum urea under dehydration than goats, reflecting differences in renal efficiency. The ability of goats to maintain renal perfusion and glomerular filtration under conditions of limited water availability may minimize increases in serum urea concentration. Creatinine is a byproduct of muscle metabolism and is excreted primarily by the kidneys. It is commonly used as an indicator of renal function. Creatinine is produced from creatine phosphate, an energy storage molecule in muscle. The rate of creatinine production is relatively constant and proportional to muscle mass. Because creatinine is freely filtered by the glomeruli and is neither reabsorbed nor secreted to a significant extent by renal tubules, its serum concentration reflects glomerular filtration rate. Reduced filtration results in increased serum creatinine. Normal serum creatinine concentrations in small ruminants typically range from 0.8 to 1.5 milligrams per deciliter. Elevated creatinine concentrations may indicate reduced glomerular filtration rate and dehydration-induced renal stress. Creatinine is considered a more specific indicator of glomerular filtration than urea because its production rate is less affected by dietary factors. However, both parameters are influenced by hydration status. The ratio of urea to creatinine can provide additional diagnostic information. Disproportionate elevation of urea relative to creatinine suggests prerenal azotemia, often due to dehydration, whereas proportional increases suggest intrinsic renal disease. Goats may maintain lower creatinine levels under similar conditions, suggesting greater renal adaptability. The consistent pattern of more stable renal function markers in goats compared to sheep reinforces the concept of superior adaptive capacity in goats under environmental stress. We now move on to the results section. The results section of the study by Chulu et al. 2025 presents the observed values for thermoregulatory and biochemical parameters measured in indigenous sheep and goats consuming naturally saline water. Let us first examine the thermoregulatory parameters reported. The thermoregulatory parameters reported in the study include rectal temperature, respiratory rate, and pulse rate. These parameters were measured under field conditions and reflect the animal's physiological responses to ambient temperature and saline water intake. The mean rectal temperature recorded for sheep consuming naturally saline water fell within the expected physiological range for adult sheep. While slight elevations were observed, these values did not exceed established thresholds for hypothermia. The recorded rectal temperature values indicate that sheep were able to maintain core body temperature within acceptable limits despite exposure to saline water and environmental heat. It is important to note that minor increases in rectal temperature may reflect increased metabolic activity, reduced evaporative cooling efficiency, or elevated ambient temperatures during measurement periods. The specific numerical values reported in the study indicate rectal temperatures for sheep drinking saline water were approximately 39.4 degrees Celsius. This value, while elevated relative to the lower end of the normal range, remains within the upper portion of typical values for healthy sheep. Context is important in evaluating whether an elevation of, for example, 0.2 to 0.4 degrees Celsius above baseline represents a meaningful physiological response or falls within normal biological variation. Statistical significance does not always equate to biological significance, particularly when sample sizes are large enough to detect very small differences. Goats exhibited rectal temperature values that were comparable to, and in some cases slightly lower than, those observed in sheep. These findings are consistent with the generally higher heat tolerance attributed to goats. The ability of goats to maintain stable rectal temperatures under saline water intake suggests effective thermoregulatory mechanisms, potentially including enhanced heat dissipation and behavioral adaptations. The mean rectal temperature for goats drinking saline water was reported as approximately 38.9 degrees Celsius. The difference between this value and the corresponding value for sheep, while statistically significant, amounts to approximately half a degree Celsius. To place this difference in perspective, diurnal variation in rectal temperature within individual animals typically exceeds this magnitude. Rectal temperature is typically lowest in the early morning hours and highest in late afternoon, with differences of 1 to 2 degrees Celsius being common. Therefore, while the species difference is real and reproducible, its practical significance must be evaluated in light of normal physiological variation. When comparing sheep and goats, the differences in rectal temperature were relatively small but consistent with prior literature. Goats demonstrated marginally better thermal stability, while sheep showed slightly higher mean values. These differences, while modest, support the hypothesis that goats possess greater adaptive capacity under combined thermal and salinity stress. The consistency of species differences across multiple studies and geographic regions lends credence to the conclusion that these represent real biological differences rather than artifacts of measurement or sampling. It should be noted that the magnitude of species differences may vary depending on environmental conditions. Under mild conditions with minimal thermal stress, differences between species may be negligible. Under severe conditions, differences may be more pronounced. The study by Chulu et al. Was conducted under naturally occurring field conditions, meaning that the thermal environment varied over the course of the study period. Measurements taken during cooler periods may show smaller species differences than those taken during peak heat. This temporal variability in environmental conditions is both a challenge and an opportunity for field research. It introduces variability that complicates statistical analysis but also allows assessment of animal responses across a range of conditions that animals actually experience. Sheep exhibited respiratory rates that were elevated relative to resting baseline values. This increase is indicative of a reliance on respiratory evaporative cooling as a thermoregulatory strategy. The observed respiratory rate suggests that sheep were experiencing a degree of thermal load, necessitating increased ventilation to dissipate heat. The specific values reported indicate that sheep drinking saline water had mean respiratory rates of approximately 39.2 breaths per minute. This falls within the normal range for sheep, but toward the upper end, suggesting activation of thermoregulatory mechanisms. Panting, as previously discussed, enhances evaporative heat loss but imposes energetic costs and may disturb acid-base balance if prolonged. The observation of elevated but not extreme respiratory rate suggests that sheep were employing panting as a thermoregulatory strategy without progressing to severe respiratory alkalosis. The fact that respiratory rates were elevated indicates that other thermoregulatory mechanisms, such as peripheral vasodilation and behavioral strategies, were insufficient to fully compensate for thermal load, necessitating increased reliance on evaporative cooling. Goats also demonstrated increased respiratory rates. However, the magnitude of increase was generally lower than that observed in sheep. This finding aligns with the notion that goats may dissipate heat more efficiently or experience less overall thermal stress. The relatively moderate respiratory rates in goats indicate effective physiological compensation without excessive respiratory effort. Goats drinking saline water exhibited mean respiratory rates of approximately 30.4 breaths per minute. This value is within the normal resting range and substantially lower than the corresponding value for sheep. The lower respiratory rate in goats could reflect several underlying mechanisms. Goats may have more efficient cutaneous evaporative cooling, reducing their reliance on respiratory heat dissipation. Alternatively, goats may experience lower metabolic heat production or may engage in behavioral thermoregulation more effectively than sheep. Behavioral thermoregulation might include seeking shade during the hottest parts of the day, reducing activity levels, or adjusting posture to maximize surface area for heat dissipation. While the study did not systematically quantify behavioral differences between species, such differences could contribute to the observed variation in physiological parameters. In direct comparison, sheep consistently exhibited higher respiratory rates than goats. This pattern suggests greater reliance on panting in sheep and potentially higher susceptibility to heat stress. The repeated observation of this trend across measurements reinforces its relevance. The difference in mean respiratory rate between sheep and goats was approximately 8.8 breaths per minute. While this might seem modest, it represents a proportional difference of approximately 29%, which is physiologically meaningful. Higher respiratory rates in sheep indicate greater energy expenditure on thermoregulation, which diverts resources from productive functions such as growth or reproduction. Over extended periods, this could translate to reduced productivity in sheep relative to goats under conditions of thermal stress. Additionally, the greater reliance on panting in sheep suggests that their capacity for other cooling mechanisms may be more limited. This could make sheep more vulnerable to heat stress under conditions where evaporative cooling is constrained, such as environments with high humidity. Sheep displayed pulse rates that were within the normal physiological range but showed slight elevations. These increases may reflect increased cardiovascular demand, peripheral vasodilation associated with heat dissipation, or mild dehydration effects. The mean pulse rate for sheep drinking saline water was reported as approximately 85.4 beats per minute. This falls within the normal range of 70 to 90 beats per minute, but toward the upper end. Elevated pulse rate under heat stress serves multiple purposes. First, increased cardiac output facilitates delivery of metabolically generated heat from the body core to the skin surface, where it can be dissipated to the environment. Second, increased blood flow to cutaneous beds supports evaporative cooling from the skin surface. However, increased pulse rate comes at an energetic cost. The myocardium must perform more work, requiring increased oxygen and nutrient delivery to cardiac muscle. Chronically elevated pulse rate could potentially lead to cardiac fatigue, though this is unlikely under moderate stress of short duration. Dehydration, if present, exacerbates cardiovascular strain by reducing blood volume. When blood volume decreases, venous return to the heart diminishes, reducing stroke volume. To maintain cardiac output in the face of reduced stroke volume, heart rate must increase proportionally. Goats exhibited pulse rates that were generally stable and comparable to baseline values. This stability suggests efficient cardiovascular adaptation under saline water intake. The mean pulse rate for goats was approximately 82.9 beats per minute. This value is within the normal range and does not indicate cardiovascular strain. The stability of pulse rate in goats suggests that their cardiovascular systems were not unduly challenged by the combination of thermal stress and saline water intake. This may reflect more effective thermoregulation through other mechanisms, reducing the cardiovascular burden. Alternatively, goats may have more efficient cardiovascular systems that can meet thermoregulatory demands without substantial increases in heart rate. Differences in cardiac stroke volume, vascular compliance, or autonomic regulation could all contribute to this species difference. The comparison of pulse rates between species reveals that sheep tend to exhibit higher values, whereas goats maintain more consistent heart rates. This difference may reflect species-specific cardiovascular responses to environmental stress. The difference in mean pulse rate between sheep and goats was approximately 2.5 beats per minute. While this difference is statistically significant, its biological significance is less clear than for some other parameters. Heart rate varies substantially within individuals over short time periods in response to factors such as movement, excitement, and digestive processes. A difference of 2 to 3 beats per minute is small relative to this within individual variation. Nonetheless, the consistency of the species difference across animals and measurement occasions suggests it represents a real phenomenon rather than random variation. Let us examine the biochemical results. The biochemical results presented by Chulu et al. 2025 provide quantitative evidence of the internal physiological adjustments occurring in indigenous sheep and goats consuming naturally saline water. These results are reported as mean values with measures of variation and are intended to reflect the animal's metabolic and electrolyte status under pastoral conditions. In this lecture, each biochemical parameter will be discussed independently, first for sheep, then for goats, followed by a comparison between the two species. The transition from thermoregulatory to biochemical results represents a shift from external indicators of physiological status to internal markers that require invasive sampling. While rectal temperature, respiratory rate, and pulse rate can be assessed through observation and simple measurement devices, biochemical parameters require blood collection and laboratory analysis. This difference has practical implications for the feasibility of monitoring in pastoral settings. Thermoregulatory parameters can be assessed frequently with minimal equipment, while biochemical parameters require specialized facilities and cannot be measured as often. Nonetheless, biochemical measurements provide unique insights into physiological status that cannot be inferred from external observations alone. The combination of thermoregulatory and biochemical data provides a more comprehensive assessment than either alone. Sheep consuming saline water exhibited serum sodium concentrations that were elevated relative to typical baseline values, yet generally remained within physiologically tolerable limits. This elevation reflects the direct intake of sodium ions from drinking water. The observed sodium concentrations suggest that sheep were able to regulate extracellular sodium through renal excretion, albeit with some degree of physiological effort. The specific values reported indicate mean serum sodium concentration in sheep drinking saline water of approximately 124.41 millimoles per liter. This value falls within the normal reference range of 135 to 150 millimoles per liter, though interestingly toward the lower end rather than the higher end as might have been predicted. This somewhat unexpected finding requires careful interpretation. One possibility is that increased water intake in response to elevated sodium load diluted plasma sodium despite increased total body sodium. Alternatively, efficient renal sodium excretion may have prevented substantial accumulation. It should also be noted that the comparison group, sheep drinking fresh water, had even lower mean sodium values, indicating that elevation in the saline water group was relative rather than absolute. Goats showed serum sodium concentrations that were similar to or slightly higher than those observed in sheep. Despite consuming the same saline water sources, goats appeared to maintain effective sodium regulation. This finding suggests enhanced renal handling of sodium and more effective osmoregulatory mechanisms in goats. The mean serum sodium concentration reported for goats drinking saline water was approximately 138.10 millimoles per liter. This value is within the normal range and higher than the corresponding value for sheep. The higher sodium concentration in goats, despite evidence of superior adaptation, may seem paradoxical. However, it likely reflects the fact that goats can tolerate higher sodium loads without adverse effects rather than that they experience greater sodium accumulation. In other words, the set point for sodium homeostasis in goats may be slightly higher than in sheep, allowing goats to maintain adequate hydration while carrying a somewhat higher sodium burden. When sheep and goats were compared, an interesting pattern emerged. Goats exhibited higher serum sodium concentrations than sheep when consuming saline water. This pattern was statistically significant and consistent. The difference, while not extreme, was consistent and repeatable. The species difference in serum sodium concentration was approximately 13.7 millimoles per liter, with goats having higher values. This represents a proportional difference of approximately 11%. The interpretation of this finding must consider the total physiological context. Higher sodium concentration in goats was not accompanied by evidence of greater physiological stress based on other parameters. This suggests that goats can tolerate higher sodium concentrations without experiencing the adverse effects that might occur in sheep at similar concentrations. This pattern is consistent with the broader theme of greater stress tolerance in goats. Rather than responding to saline water by minimizing sodium absorption, goats appear to accommodate higher sodium loads while maintaining effective physiological function through other adjustments. Serum potassium concentrations in sheep remained within the normal physiological range. Minor fluctuations were observed, but no dramatic deviations were recorded. These findings indicate that potassium homeostasis was largely maintained despite saline water intake. The maintenance of normal potassium concentrations is important because potassium imbalances can have serious consequences for cardiac and skeletal muscle function. The fact that potassium remained within normal limits suggests that animals were not experiencing severe metabolic derangements. Potassium homeostasis is maintained through a balance of dietary intake, cellular uptake and release, and renal excretion. The observation of stable potassium concentrations indicates that these regulatory mechanisms were functioning adequately. Goats demonstrated similarly stable potassium concentrations. The values recorded were comparable to those of sheep, suggesting that potassium balance was not substantially disrupted in either species. The similarity of potassium concentrations between species and between water types indicates that this parameter was not a major point of differentiation in the present study. This does not mean that potassium homeostasis is unimportant, only that saline water intake did not substantially challenge potassium regulation in either species under the conditions of this study. The comparison between species revealed minimal differences in potassium concentrations. Both sheep and goats maintained potassium levels within expected ranges, indicating that saline water consumption had limited impact on this parameter. The lack of significant differences in potassium concentration between groups simplifies interpretation in some respects, as it eliminates potassium as a major variable in explaining other physiological responses. However, the measurement of parameters that do not differ among groups is still valuable, as it helps to characterize the full physiological profile and ensures that important derangements are not being overlooked. Sheep exhibited increased serum chloride concentrations, reflecting the chloride content of the saline water. These elevations were modest and did not exceed physiological tolerance thresholds. The observed chloride levels suggest effective renal excretion, albeit with increased solute load. Chloride, as the primary anion accompanying sodium in saline water, necessarily increases when sodium increases. The parallel changes in sodium and chloride maintain electroneutrality. The fact that chloride increases were modest indicates that renal excretion was sufficient to prevent extreme accumulation. However, the elevation nonetheless represents an increased burden on renal function. Goats maintained serum chloride concentrations that were comparable to or slightly lower than those of sheep. This again indicates a greater capacity for electrolyte regulation. The lower chloride concentrations in goats relative to their sodium concentrations might seem contradictory, as these ions are typically regulated in parallel. However, measurement variability and differences in the anion gap could account for this pattern. As with sodium, goats exhibited more stable chloride concentrations than sheep. The repeated observation of this trend across electrolyte parameters reinforces the notion of species-specific adaptive differences. The pattern of more stable electrolyte concentrations in goats is consistent across multiple ions, suggesting a generalized superiority in ionic homeostasis rather than specific adaptation to particular ions. Sheep showed slightly elevated serum total protein concentrations. This elevation may be indicative of mild hemoconcentration, potentially associated with reduced water intake or increased water loss. The increase in total protein was modest but consistent. Hemoconcentration, as indicated by elevated total protein, suggests that plasma volume may have been reduced in sheep drinking saline water. This could result from inadequate water intake due to poor palatability of saline water, from increased water losses through evaporative cooling, or from a combination of both. The magnitude of increase in total protein provides a rough index of the degree of hemoconcentration. Large increases would suggest substantial dehydration, while modest increases indicate mild volume contraction. Goats exhibited total protein concentrations that were generally lower and more stable. This suggests better hydration status or more efficient water utilization. The stability of total protein in goats indicates maintenance of plasma volume despite saline water intake. This could reflect greater voluntary water intake, reduced evaporative water losses, or more efficient water conservation through renal mechanisms. The difference in total protein between sheep and goats suggests that goats may experience less dehydration under saline water intake, or may compensate more effectively. The pattern of elevated total protein in sheep relative to goats is consistent with the interpretation that sheep experience greater challenges to hydration status under saline water consumption. This finding integrates with thermoregulatory results showing higher respiratory rates in sheep, as increased respiratory evaporative cooling would increase water losses and contribute to dehydration. Sheep displayed serum glucose concentrations within normal physiological ranges. Slight reductions were observed in animals consuming saline water, potentially reflecting reduced feed intake or altered energy metabolism under stress. The specific values indicated that sheep drinking saline water had mean glucose concentrations of approximately 63.33 mg per deciliter, compared to approximately 71.33 mg per deciliter in sheep drinking fresh water. This reduction of approximately 8 mg per deciliter represents a proportional decrease of approximately 11%. While statistically significant, both values remain within the normal range, suggesting that glucose homeostasis was maintained despite the challenge. Reduced glucose concentrations could reflect decreased feed intake if saline water affected palatability or if animals spent less time grazing due to thermal stress. Alternatively, stress-induced alterations in hormonal regulation of glucose metabolism could contribute. Goats maintained glucose concentrations that were stable and similar to those of sheep. No marked hypoglycemia or hyperglycemia was observed. The stability of glucose concentrations in goats across water types suggests that energy metabolism was not substantially perturbed by saline water consumption. This could indicate maintained feed intake and effective metabolic regulation. The comparison revealed minimal differences between species, suggesting that glucose metabolism was not substantially affected by saline water intake in either sheep or goats. While both species showed reductions in glucose when drinking saline water compared to fresh water, the magnitude of reduction was similar, indicating that this was not a point of species-specific differential response. The parallel response of both species to saline water with respect to glucose suggests that this effect may be mediated through mechanisms common to both species, such as reduced feed intake or thermal stress, rather than through species-specific physiological differences. Sheep exhibited increased serum urea concentrations when consuming saline water. This elevation may reflect reduced renal clearance associated with dehydration or increased protein catabolism. The mean serum urea concentration in sheep drinking saline water was approximately 69.72 mg per deciliter, compared to approximately 68.96 mg per deciliter in those drinking fresh water. This small difference, while statistically significant given the sample size, is very modest in absolute terms. Both values fall within the normal reference range, indicating that kidney function was adequate. Nonetheless, the direction of change is consistent with mild pre-renal azotemia, wherein reduced renal perfusion due to dehydration decreases urea clearance, leading to accumulation in blood. Goats showed serum urea concentrations that were similar to those of sheep. Values were elevated in animals drinking saline water relative to fresh water, with goats showing mean concentrations of approximately 74.42 mg per deciliter when drinking saline water. Interestingly, goats exhibited higher absolute urea concentrations than sheep in both water treatment groups. This species difference could reflect differences in protein metabolism or in renal handling of urea. The comparison revealed that goats had higher serum urea concentrations than sheep regardless of water source. This pattern is statistically significant and indicates a consistent species difference. The higher urea in goats is somewhat unexpected given their apparent superior adaptation to saline water based on other parameters. However, elevated urea in the absence of elevated creatinine suggests that this reflects metabolic differences rather than impaired renal function. Goats may have higher rates of protein turnover or may rely more heavily on gluconeogenesis from amino acids, both of which would increase urea production. As long as renal function remains adequate to excrete the increased urea load, this need not indicate pathology. Sheep displayed serum creatinine concentrations that were elevated in animals drinking saline water. Mean values were approximately 3.03 mg per deciliter. This value is at the upper end of or slightly above the normal reference range of 0.8 to 1.5 mg per deciliter, suggesting possible mild renal stress. Elevated creatinine indicates reduced glomerular filtration rate, which could result from dehydration reducing renal blood flow or from direct effects on kidney function. Goats maintained similar creatinine concentrations to sheep, with mean values of approximately 3.13 mg per deciliter in animals drinking saline water. As with urea, both species showed elevations in creatinine when drinking saline water, but the elevations were modest and did not indicate severe renal dysfunction. The comparison revealed minimal differences between species in creatinine concentration, with both showing modest elevations. This pattern is consistent with mild renal stress affecting both species similarly. The fact that both species showed parallel increases in creatinine suggests that the renal challenges imposed by saline water affected both species to similar degrees, despite apparent differences in other aspects of their physiological responses. We now transition to the discussion. The discussion section of the study by Chulu et al. 2025 seeks to contextualize the observed thermoregulatory and biochemical responses of indigenous sheep and goats within the broader body of literature on saline water intake, heat stress, and small ruminant physiology. In doing so, it reiterates many of the findings already presented in the results section, while offering cautious interpretations grounded in established physiological principles. In field-based studies such as this one, the discussion must carefully balance interpretation with recognition of the constraints inherent in observational research. Causal claims must be tempered by acknowledgement that alternative explanations may exist. Let us first examine the interpretation of thermoregulatory responses. The maintenance of relatively stable rectal temperatures in both sheep and goat suggests that indigenous small ruminants possess effective thermoregulatory mechanisms that allow them to cope with combined environmental heat and saline water intake. Rectal temperature, as previously discussed, serves as a proxy for core body temperature. The absence of extreme hypothermia indicates that the animals were able to balance heat production and heat loss. The observation that rectal temperatures remained within or near normal limits is reassuring from an animal welfare perspective, as it indicates that animals were not experiencing severe heat stress that could compromise health or survival. However, the modest elevations observed, particularly in sheep, suggest that thermoregulatory mechanisms were being actively employed to prevent further temperature increases. The animals were in a state of thermal challenge, requiring physiological compensation to maintain homeostasis. The fact that compensation was successful under the conditions of this study does not guarantee that it would remain successful under more extreme conditions or over longer time periods. Chronic activation of thermoregulatory mechanisms may impose cumulative costs on productivity and health. The elevated respiratory rates observed, particularly in sheep, can be interpreted as a compensatory response aimed at increasing evaporative heat loss. Panting increases airflow across moist respiratory surfaces, facilitating heat dissipation. However, reliance on increased respiratory rate may come at a physiological cost, including increased energy expenditure and potential alterations in acid-base balance. The lower respiratory rates observed in goats suggest more efficient thermoregulation or reduced thermal burden. The energetic cost of panting is not trivial. The respiratory muscles must perform mechanical work to move air in and out of the lungs, and this work generates heat as a byproduct. Under extreme conditions, the heat generated by respiratory muscle activity could potentially approach the heat dissipated through evaporative cooling, limiting the net benefit of panting. Additionally, rapid breathing associated with panting may reduce the efficiency of gas exchange if tidal volume decreases substantially. Very shallow breaths primarily ventilate the anatomic dead space of the airways without contributing significantly to alveolar ventilation. These considerations suggest that while panting is an effective short-term thermoregulatory mechanism, prolonged reliance on it may be unsustainable. Pulse rate provides insight into cardiovascular adjustments under stress. The modest increases observed in sheep may reflect increased circulatory demands associated with heat dissipation and electrolyte regulation. Goats, by contrast, exhibited more stable pulse rates, suggesting that cardiovascular homeostasis was maintained with less physiological strain. The cardiovascular system plays a central role in thermoregulation through its control of heat distribution within the body and heat exchange with the environment. Redistribution of cardiac output to favor cutaneous circulation facilitates heat transfer from core to periphery. However, this redistribution must be balanced against the need to maintain adequate perfusion of vital organs. The cardiovascular system has finite capacity, and extreme demands for both thermoregulation and other functions may create conflicts. The stability of pulse rate in goats suggests that their cardiovascular systems were operating within their capacity without approaching limits. In sheep, modest pulse rate elevation indicates greater cardiovascular demand, though not to a degree suggesting imminent cardiovascular failure. Thermoregulatory responses cannot be considered in isolation from hydration status. Saline water intake influences plasma osmolality, which in turn affects thirst, urine concentration, and evaporative cooling capacity. The combined assessment of thermoregulatory and biochemical parameters provides a more complete picture of physiological adaptation. The interdependence of thermoregulation and hydration creates potential for positive feedback loops that can exacerbate stress. High temperatures increase evaporative water losses, increasing water requirements. If water intake is limited due to poor palatability of saline water, dehydration develops, which impairs evaporative cooling capacity, which leads to further temperature elevation. Breaking this cycle requires either improved water availability and quality, behavioral adaptations to reduce heat load, or physiological mechanisms that enhance water conservation. Indigenous breeds adapted to arid environments may possess all three types of adaptation. Behavioral knowledge of where to find the best water sources, selective feeding on moisture-rich vegetation, and physiological capacity for water conservation all contribute to survival. We shall now move to the interpretation of serum sodium and chloride findings. The elevated serum sodium and chloride concentrations observed, particularly in goats, are consistent with the ingestion of saline water. These findings align with previous studies reporting similar responses in small ruminants exposed to high salinity water sources. The ability of both species to maintain electrolyte concentrations within tolerable limits suggests adequate renal excretory function. The kidneys serve as the primary regulators of sodium and chloride balance, adjusting excretion to match intake. When intake increases, healthy kidneys respond by increasing excretion, thereby preventing excessive accumulation. The fact that serum concentrations were elevated but not at dangerous levels indicates that renal regulation was functioning, but that the rate of excretion did not perfectly match the rate of intake, resulting in a new steady state at slightly elevated concentrations. This is analogous to filling a bathtub while the drain is open. If inflow exceeds outflow capacity, the water level rises until it reaches the overflow drain, at which point a new equilibrium is established at a higher level. Let us look at renal function indicators, urea and creatinine. Serum urea and creatinine are commonly used indicators of renal function and hydration status. The elevations observed in both species may indicate reduced glomerular filtration associated with dehydration or increased metabolic load. The fact that elevations were modest and both parameters increased proportionally suggests prerenal azotemia rather than intrinsic kidney disease. Prerenal azotemia refers to elevation of nitrogenous waste products in blood due to factors external to the kidneys, most commonly reduced renal perfusion due to dehydration. This is distinguished from renal azotemia, wherein elevations reflect damage to kidney tissue itself, and postrenal azotemia, wherein obstruction prevents urine excretion. In prerenal azotemia, kidney function is inherently normal, and concentrations of waste products normalize once perfusion is restored. This contrasts with renal azotemia, wherein structural damage may permanently impair function. The reversibility of prerenal changes makes them less concerning from a long-term health perspective, provided that adequate rehydration can be achieved. The slight elevation in serum total protein observed in sheep suggests hemoconcentration, likely resulting from reduced plasma volume. This finding is consistent with mild dehydration. In goats, more stable total protein concentrations indicate better maintenance of plasma volume. Total protein concentration is a relatively crude indicator of hydration status, but has the advantage of being measurable on most clinical chemistry analyzers without additional specialized equipment. More sophisticated methods for assessing hydration include measurement of plasma osmolality, which directly quantifies the concentration of osmotically active particles, and assessment of urine-specific gravity, which indicates the concentrating ability of the kidneys. However, these methods require specialized instruments not always available in field settings, making total protein a practical alternative. The relative stability of serum glucose concentrations in both species suggests that energy metabolism was not severely disrupted. This stability may reflect effective hormonal regulation under environmental stress. The maintenance of glucose homeostasis is critical for supporting brain function and other glucose-dependent tissues. The brain relies almost exclusively on glucose as an energy substrate and cannot readily utilize alternative fuels. Therefore, preservation of blood glucose concentration is a high homeostatic priority, and multiple redundant mechanisms exist to maintain it. These include hepatic gluconeogenesis, glycogen mobilization, and hormonal regulation of glucose utilization by peripheral tissues. The fact that glucose concentrations remain stable despite other physiological challenges indicates that these regulatory mechanisms were functioning effectively. The findings of this study are consistent with previous research conducted in arid and semi-arid regions. Multiple studies have reported that goats exhibit greater tolerance to saline water than sheep, as evidenced by more stable physiological and biochemical parameters. Such consistency across studies strengthens confidence in the observed patterns. Scientific confidence increases when independent studies employing different methodologies in different locations with different animal populations arrive at similar conclusions. This convergence of evidence suggests that the observed patterns reflect robust biological phenomena rather than artifacts of particular study designs. However, it is also important to note sources of variation among studies. Different studies may use different levels of water salinity, different environmental conditions, different breeds of animals, and different measurement protocols. These differences can lead to quantitative variation in results even when qualitative patterns are similar. Meta-analysis, which statistically combines results from multiple studies, can help to quantify the magnitude and consistency of effects across diverse conditions. From a practical perspective, the findings suggest that goats may be better suited to environments where access to fresh water is limited. Sheep, while capable of tolerating saline water to some extent, may experience greater physiological strain. These implications are particularly relevant for pastoral communities facing increasing water scarcity. Livestock species selection is a key decision in pastoral production systems. The choice of which species to raise must consider multiple factors, including market demand for different products, cultural preferences, forage availability, disease resistance, and environmental adaptability. In regions where water quality is marginal, the superior salt tolerance of goats may tip the balance in favor of goat production. However, this advantage must be weighed against other considerations such as the relative market value of goat versus sheep products. In some contexts, mixed species herds including both sheep and goats may offer advantages through portfolio diversification. Different species may perform better under different conditions or in different years, reducing overall risk. The discussion acknowledges several limitations, including variability inherent in field-based studies, lack of controlled experimental conditions, and potential confounding effects of diet and activity level. Despite these limitations, the study provides valuable insights into real-world adaptation. Let us transition to the conclusions section. The conclusion section serves to summarize the findings of the study conducted by Chulu et al. 2025, reiterate their significance, and outline broader implications for livestock management under pastoral systems. In keeping with the tone of this lecture, these conclusions will be presented slowly, repetitively, and with extensive restatement of previously discussed material. The primary objective of the study was to evaluate the thermoregulatory and biochemical responses of indigenous sheep and goats consuming naturally saline water under pastoral conditions in the Mid-Rift Valley of Ethiopia. This objective was addressed through the measurement of rectal temperature, respiratory rate, pulse rate, and serum biochemical parameters related to electrolyte balance and renal function. Both sheep and goats demonstrated the capacity to maintain rectal temperature within physiologically acceptable limits. This indicates that indigenous small ruminants possess effective mechanisms for thermoregulation, even under conditions of elevated ambient temperature and saline water intake. However, sheep consistently exhibited slightly higher rectal temperatures, higher respiratory rates, and modestly elevated pulse rates. Goats, by contrast, demonstrated greater stability across these parameters. The pattern of species differences in thermoregulatory parameters creates a clear picture of differential adaptive capacity. While both species can tolerate the environmental conditions of the study area, they do so through different physiological strategies and with different degrees of effort. The biochemical analyses revealed that saline water intake influenced serum electrolyte concentrations and renal function indicators. Notably, serum sodium concentrations differed between species, with goats showing higher values but apparent tolerance. Serum urea and creatinine showed modest elevations in both species, and total protein concentration suggested mild hemoconcentration, particularly in sheep. Goats consistently maintained more stable biochemical profiles overall. The biochemical findings complement and extend the thermoregulatory results, providing insight into internal physiological status that is not apparent from external observation. When considered collectively, the thermoregulatory and biochemical data indicate that goats are more resilient to the combined challenges of saline water intake and environmental heat stress. Sheep, while capable of tolerating these conditions, appear to do so with greater physiological effort. The integration of multiple lines of evidence strengthens conclusions beyond what could be drawn from any single parameter. The consistency of patterns across diverse measurements creates a compelling case for species-specific differences in adaptation. The findings of this study have practical implications for pastoral systems in arid and semi-arid regions. In areas where access to fresh water is limited, goats may represent a more suitable livestock species due to their superior tolerance to saline water. This does not imply that sheep cannot be raised under such conditions, but rather that additional management considerations may be required. Livestock management recommendations should be evidence-based, drawing on research findings while remaining sensitive to local context and constraints. What works in one setting may not be optimal in another. For the Mid-Rift Valley of Ethiopia and similar regions, the evidence suggests that goat production may offer advantages under conditions of water stress. However, implementation of this recommendation must consider market factors, cultural preferences, and availability of other resources. Climate change is expected to exacerbate water scarcity and increase the salinity of available water sources in many regions. Studies such as this provide valuable information for adapting livestock production systems to changing environmental conditions. The demonstrated resilience of goats suggests that they may play an increasingly important role in future pastoral livelihoods. Climate adaptation in agricultural and pastoral systems requires forward-looking planning based on projections of future conditions. Research on current responses to environmental stressors provides a foundation for predicting how systems will respond to intensified stressors. If current conditions with moderately saline water already reveal differential stress between species, it is reasonable to expect that future conditions with higher salinity will show even greater differentiation. Proactive adjustment of production systems in anticipation of changing conditions may be more successful than reactive responses after problems have already developed. The study recommends further research to assess long-term effects of saline water intake on productivity and reproduction, evaluate performance under varying levels of water salinity, explore genetic and physiological mechanisms underlying adaptation, and investigate management interventions that might enhance tolerance in sheep. In closing, the study by Chulu et al. 2025 contributes valuable empirical data to the literature on livestock adaptation under pastoral conditions. Its findings reinforce existing knowledge while providing region-specific insights relevant to Ethiopia's Mid-Rift Valley. Thank you for your attention, or alternatively, your restful repose during this session of the Boring Lecture Series. We hope this has been a useful resource for your studies or a supportive environment for your rest. This lecture was narrated by Dr. Owen On, an artificial voice for the Boring Lecture Series. Please see the description for further resources and the complete citation to the primary literature discussed throughout this presentation. If you found this level of detail sufficient, please consider subscribing for our weekly sessions. We will now fade to soothing bioacoustic sounds and ambient tones to allow for a period of quiet reflection or continued rest. Good night.